Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome everybody back to In the Interim. I'm your host, Scott Barry. I'm your co-host, Scott Barry. My co-host, my common co-host, Kert Viele, is back. Though, Kert, it's been a bit of a delay. Bit of a gap.
Happy to be here. Happy to be here.
We may find out if Kert has any pet peeves for today. But let's get to the topic of the day. And, we are somewhere in the 60 plus episodes, Kert, and we have not done an episode on response adaptive randomization. Something that makes people passionate. It's controversial. It's a great topic for innovative trial designs. I think it's especially a great topic as the world of AI is coming. The world of clinical trials is evolving. What is the role of response adaptive randomization in it? What are our thoughts on it? Lots of critics of response adaptive randomization. So let's get to the bottom, to the top, to the sides of response adaptive randomization.
Sounds great.
Okay, so it's a controversial topic. Let's start with what is response adaptive randomization. Remember, my wife loves to tune in on these, and she's going to want to make sure that everybody understands the topic. So explain to Tammy what response adaptive randomization is.
All right. First off, hi, Tammy. Good to see you. So the idea behind RAR, response adaptive randomization, we'll abbreviate it from here on out, is essentially all adaptive trials. They do interim analyses, and they make some kind of adjustment to the trial at those interim analyses. In RAR, the adjustment that you're making is to change the allocation probabilities. And usually what we would do is we would increase allocation to arms that are performing better. We decrease allocation to arms that are performing worse. The basic idea is to do a couple things. One is to try to treat patients better in the trial, try to get them the better performing arms. And we hope we get some better inferences at the same time. But I know we're going to touch on that when we get into some of the controversies, because that sometimes happens and sometimes doesn't.
Yeah. So examples of how this may be. And we've been involved in multiple trials where response adaptive randomization has been used. And so a couple of those, the BAN 2401 trial was a trial of lecanemab, which is now approved for Alzheimer's disease. Their phase 2 trial was a placebo and 5 active arms of lecanemab. It was actually 2 frequencies and 3 doses. And those 5 arms, they did 196 patients enrolled, where it was initially fixed randomization to those 6 arms in the trial. An interim analysis took place, and a new randomization probability was set for each of the arms. Essentially, the control was a fixed rate. It was slightly different than that, but for all intents and purposes, the control was fixed. And the other 5 could vary. With the remaining probability, a new randomization vector was created. For the next period of time, patients were randomized using those probabilities. And then 50 patients later enrolled, a new interim was done, and this was repeated during the course of the trial. And it ended up that 2 of those doses, the 2 higher doses with the 2 frequencies, were the most patients explored. One of them was moved to phase 3, and it was a successful in a phase 3 trial. The goal of that phase 2 trial was to find the best dose. What is the best arm, and how good is that arm? And we'll come back to whether RAR is good or not, but that was an example of response adaptive randomization being used. I'll point out one more. We'll probably talk about other trials and how they do it and the good and the bad of it. I'll touch on another one just because I've done 2 recent podcasts on it, and we are doing a podcast on the results of the ICECAP trial. By the way, it's been recorded, and we're just waiting for JAMA to publish the article, and then we're going to release the discussion of the ICECAP trial. The ICECAP trial had 10 arms. All arms were the duration of hypothermic cooling for the treatment of cardiac arrest, post-cardiac arrest, resuscitation. You go to the emergency room, and they do different durations of hypothermic cooling. And the goal was to find the best duration. And largely, was there an increasing dose response? Largely, what is the best duration was the goal of that. And there were 10 arms in that trial.
And you should also add that the lecanemab story, you published that article as well, so people can go back and read that and see what happened interim to interim. I thought that was, a lot of times we don't publish that kind of information. I thought it was good that it's out there.
Yeah, it's the only trial I know of that published the actual interim reports that were done at each of, I think there were 18 or 19 interim analyses. And it shows the quantities that were used to calculate the randomization probabilities. It shows what each one were at all the interims. You can see those reports. Fantastic. I'm trying to get the ICECAP people to publish that, and we will. We'll show each of the interims for that as well, so people can follow along. Watch the movie at home of the trial. So that gives a little bit of a flavor in just explaining response adaptive randomization. Now, to some extent, it sounds fantastic. Is this? If I'm a patient, I feel like I would like to be in a trial that uses response adaptive randomization, just at face value. At the same time, within this, I think there are a lot of people who are unfamiliar with clinical trials, that when I explain to them adaptive trials, they look at me like I have 3 heads. Like, don't all trials do this? Shouldn't the allocation of treatments at the latter part of the trial be informed by the earlier part of the trial and what's given to them? And when you describe that, no, most of them not, they're surprised by that. So there's that part, but there are critics of it. And I don't know where we fall in this, Kert. I'm not pro RAR. As I don't think there's value in that. It's a tool in the trial to, in some cases, get better answers. Some cases treat patients better, but it's a tool available to us as trial designers, and it has good and bad in it. I think it's fair to say there are authors out there who are against RAR and write articles. Don't fall for the RAR trick. Don't do it. It's bad. But it is quite controversial in the literature.
At least we're very persuasive because we're laying out traps and we're getting people to do our stuff. But ironically, we don't. How many of these do we actually do? We certainly do them. I mean, we do hundreds of trials over the years, but it's certainly not 60% of these involve RAR. It's particular cases where we've done it and particular cases where we haven't.
Very much so. So let's talk about what those cases. So if what are the critics of RAR? Where does this perhaps not go well?
So, this gets at whether, we're critics of RAR for certain cases as well. So, I mean, the central thing, I think what you said from the patient standpoint, obviously, if you're enrolling late in the trial, you want to be, hey, a lot of people have generated information. I want to get the benefit of that. I would fully understand that perspective. There's a scientific aspect that the purpose of the trial is to generate information beyond the trial. If you're doing in lecanemab, there are millions of people who are suffering from Alzheimer's that want a good and effective drug. So we want to keep them in mind while we're also trying to help patients in the trial. one thing that happens is I'm not going to do a lot of math here at all, of course, but if you're looking at estimating a treatment effect, so the sample sizes in each of the arms matter. You're comparing control to treatment. If you can make both of those arms have bigger sample sizes, you're going to get better estimates. You're going to get more information. So now the question is, well, how does RAR actually do that? Well, RAR does that by increasing allocation to some arms, decreasing it to others. If you're in a 2-arm trial, for example, this is the classic, and a lot of the papers that are against RAR, they focus on 2 arms with reason to say it's problematic here. You increase the treatment allocation potentially, but you decrease the control. And this gets into kind of a robbing Peter to pay Paul situation. You can actually decrease, you estimate treatment effects worse in 2-arm RAR than you would in doing a fixed trial. So a lot of people have noted this. There's a problem. 2-arm trials, we don't do RAR very often. In this case, I think it's a really specialized issue. All that's going to be turned on its head in a multiple arm setting. And I think we'll get to that in a minute. But 2 arms is if you read the literature, 2 arms is a very, very, very controversial case. And I think we agree with the limitations there.
So a lot of critics of RAR will point out that in a 2-arm trial, moving away from 1-to-1, it reduces your power. It reduces your inferences about the difference between them. Now, there are some odd cases where it might be good to reduce one. The variability is different in the 2 arms and all of that. But largely, you reduce power in that. And then they make a sweeping argument so you should never do RAR.
Yeah, so that's my pet peeve. You wanted a pet peeve.
There we go.
My pet peeve is how overgeneralized the RAR literature is. RAR, it's a lot of different methods. You can do it a lot of different ways. You can do it in a lot of different cases. A lot of papers are written, I'm going to explore this version of RAR in this setting, but I'm going to generalize it to everything. And that just doesn't serve us well.
And they say, look, this method doesn't work here. RAR doesn't work, and they generalize it to every possible RAR cases of that. Okay, so you talked about the 2-arm case, the 2-arm setting, and so that would be a specialized scenario where the goal isn't entirely treatment effect. Yeah. There may be other goals, and we'll come to that a little bit. So let's just take another criticism of it is temporal trends. So what is the criticism about temporal trends?
So there's something really magical about doing fixed randomization, not doing RAR. So if I run a trial and I enroll patients 1-to-1, and I have patients that enroll early in the trial, I have patients that enroll late in the trial. If I look at the treatment and control groups, however those split between how many patients came in early and how many came in late, they're the same between treatment and control because I've done 1-to-1 the whole way. I've equalized allocation the whole way through the trial. If I do RAR, that doesn't happen necessarily. If I start the trial 1-to-1 and then shift to 3-to-1, for example, or some people would say 9-to-1, which I don't think we get to very often. But in any case, if you do that, so what's going to happen is you go 3-to-1 in favor of treatment later in the trial. You look at the treatment subjects, they're more late patients. You look at the control subjects, there are fewer late patients. If there's any kind of anything going on, and the classic example is COVID, where there's a new variant coming in and people react to it differently, that is going to generate a bias. And that is potentially really, really bad. So, I mean, it depends on how big the bias is, but it can completely invalidate your conclusions. Typically, we would add terms to our model. We'd model time to address this. But that's generally what the issue is you do need to take active steps to correct for that and make sure you're robust.
So that creates biases. And that term, it creates biases, always depends on how you're analyzing the data.
Right.
If you ignore the fact that it's different over time and it's not part of the model, then it could cause bias. If you're adjusting by time, then, assuming you have additive effects across time, then it's not biased within that setting. So there are ways to adjust for time if it's a concern. There are some cases where we have much less concern about time effects. In some cases, we have larger concerns about time. So there are ways to adjust for that. Okay. There's one other potential criticism, which is the rare circumstance where people may be unblinded to what's the allocation is that you may learn one treatment is doing better than another one just by the fact it's being given more.
Yeah.
But in a blinded trial where patients are blinded, investigators are blinded, nobody would know that a particular treatment is given more. In trials where they're unblinded, this becomes also a potential criticism of RAR.
Yeah, and among many other criticisms of an unblinded trial, lots of things can happen in those settings, and RAR may make them worse.
Okay, so we've set up potential negatives. This unblinding aspect of it is a potential negative. Temporal trends is a potential negative to it, and in a 2-arm trial, you reduce power. So, okay.
It sounds awful.
Yeah, sounds bad. So what are the potential positives of RAR? We started, it seemed like a positive thing. What are the positives of RAR?
Well, so there is a case where RAR is a good match. So, and that case, the one we often use it for, is situations where we are looking at multiple arms. We're not in a two-arm setting. And we're looking for the best arm among those alternatives. So the classic examples here would be dose finding. I'm not particularly worried about, which arm is the fourth best. I want to know what is the best. In the epidemic settings, the pandemic settings, we want to know what's the best treatment for patients. We don't necessarily need to know the others. In those cases, what RAR does for you is because you are increasing allocation to the best arm, it's the one doing the best, it's the one you're going to raise, you get more information on that best arm. You want to be careful here. This is the thing we've always been telling people is you want to make sure you maintain the control allocation because that's part of your comparison. If you're going to do an estimate at the end to compare to control, you need controls. Don't skimp on those. But in those cases, RAR will give you more power because it gets more patients on the arms that matter in your comparison. It gets you lower bias, lower variance. So basically you get better estimates. All of these inferential things start working in your favor.
So in a setting, and we looked at in a potential trial for Ebola, for example, you might have 4 active treatments that could be brought in as a treatment for it. If you're increasing the probability of 1 or 2 of those treatments and keeping control the same, you have a better idea of figuring out the best. But now you've reduced the randomization to arm 4. Yeah. And in a setting, the question is, do you care about that? So power is such an it's not an odd thing, but it's a really important thing to make sure we understand what are we trying to do in the trial? What are the goals of the trial? If it's trying to identify every possible treatment that's good or bad, you might not want to do RAR because it's not finding the best. If it's about finding the best and identifying the effect, it can increase the power. And dose finding is a great example of that because you don't care about arms that aren't as effective. You're trying to identify the best. And so it's a perfect example. And it's commonly where we do it the most at Barry is a dose finding trial, where it can create a better trial design. Okay, now at the same time, so if somebody enters into a trial and there's this framework that's set up in these trials of a VIP, where suppose you were running a trial and a VIP came in and you wanted to treat the patient as well, and you could look at the data and assign one of the arms. As the trial goes on, you're more and more likely to put that patient on a better arm because you're learning and you would treat that VIP better. Now, we don't do that in trials and VIPs don't get to come in and look at the data of it. but that if we're increasing the probability to arms that are doing better and we're learning more efficiently, we're treating patients better in the trial.
Yeah.
And so a patient that comes in later in the trial is more likely to be given a more effective arm in the trial.
So we've talked about this. It probably doesn't happen in trials very often, but we've talked about this example in the context of a learning healthcare system where the whole healthcare system is trying to improve itself, but somebody walks in the door going, I don't want you to randomize me. You just look at the data and tell me which one's the best, and I want that. And you can imagine situations where we talk about the ethics of it for a long period of time, but it's at least conceivable. The nice thing about RAR is in that ethical case, if you were doing fixed randomization, that VIP coming in wanting special treatment, they actually get treated much better than the average patient because the average patient's getting randomized, not making use of information. If you're doing RAR, what's happening is the difference between that VIP and the average patient in the trial is actually pretty darn small. It's a couple, if you're doing mortality, it's a couple percent rather than 25%. So it really helps on getting patients treated well and the idea that we're still learning as well as making use of that information.
Yep. Okay, so from a perspective, and I started this off with, if I'm a patient going into a trial and there are two trials and one of them is doing RAR and one of them is not, I'd rather go in the trial with RAR. I find that a better place to go, which interestingly, we do a number of trials, especially rare disease. We're treating a pediatric disease, a Duchenne muscular dystrophy, where you might do 2-to-1 or 3-to-1 randomization. There's a very strong preference of a patient to enter into a trial where they're more likely to be put on the active arm. And it's done as a tool to increase enrollment within the trial to increase the likelihood patients would agree to enter into that trial because it's favoring the active arm. Now you've got a procedure that learns from the data and goes to different arms within it, which is a perception generally, I think, that patients would rather be in that trial.
And I think we ought to add kind of to the objections to RAR here that, we simulate these trials and how are we doing this RAR? How are we increasing the allocation? You can, of course, do this badly. When we present this to people who have never, done adaptive trials before, the natural question is, well, what if you overreact to early data and the early data is just noisy and wrong? And certainly the answer is if you do RAR badly, you can overreact to that. There are papers that have gone through, doing interims at a sample size of 3 where, hey, this isn't working out well. So you want to simulate this. You want to make sure that you do this in a way where you don't overreact, but you're making use of the information. And that requires basically a seasoned practitioner in order to find the right balance.
Yeah, so we would never enter into a trial, put an algorithm together that does RAR, and just hope it works. We simulate thousands of trials, millions of trials. We make sure under a huge range of scenarios, it's getting better answers, or it's accomplishing the goals that it's intended to improve upon. If it's that trial itself, we want to improve the outcome of patients. If it's to learn the right dose. So we simulate them extensively ahead of time. There's a famous example of an RAR algorithm where in hindsight, I'm not sure that they like the outcome of it within it, and it's the ECMO trial. Bartlett years ago ran a trial, and it was newborns that had particular respiratory issues. They were being randomized between conventional care and ECMO, using an ECMO machine for them. Now, what was the design? It was called a play the winner design. and our polio urn design, where initially there's a bowl and there's two balls in the bowl. One is red for conventional, one is blue for ECMO. And the first patient comes in, you grab a ball, and whatever ball is selected, that's assigned to the patient. If that patient is a success and they survive, survival was the endpoint, you put back the ball on the therapy they were given. So if they were given the blue therapy and they lived, you put another blue ball in the bowl. And then the next patient that comes in has a 2/3 chance of being given the blue therapy. If they're given blue and they don't survive, you put a red ball in the urn. And now it's more likely the patient would get red. And every patient that continues, and you put more and more balls in the urn in there, and therapy's doing better, have more balls in there. Therapy's doing worse, the other one has more. And it was a procedure for doing response adaptive randomization in that. Now, what happened in the trial is the first patient that came in was randomized to conventional care, and the patient died. And so a new ball was put in for ECMO. The next patient that came in got ECMO and survived. So now a new ball is put in. So now it's 75% ECMO. Well, it went on a run of like 27 consecutive ECMO patients, and they were all surviving. I think there may have been a death in there. But the trial ended up, I believe, like 26 out of 27 on ECMO and 0 for 1 or 0 for 2. I think at the end, they even added a few more conventional therapies on there. But it ended up incredibly disparate in the trial. Now, it turns out ECMO is highly effective. It's better than conventional. We know that now. But the trial was greatly criticized. And I had the benefit once of asking Bartlett, had you seen a simulation that showed what happened in the trial, would you have said that was good? I'm glad it did it. He said, no, we would have done something different. And that's the value of simulation, to be able to evaluate that particular algorithm.
So one aspect of that, so all of these play the winner, the polio schemes, they come out of a particular literature, which obviously, your father, Don's, heavily contributed to. But there is one aspect of that. A lot of that literature is based on trying to treat a sequence of patients well without having any regulatory aspect to it. So at no point do you have to generate, p less than 0.025, so to speak. One thing that people should keep in mind is that when you put that kind of regulatory requirement on it, you need a different kind of evidence. And so that's one of the things more modern RAR methods do, is they take into account the regulatory environment as well, which is a change over the past 40 years.
Which is doable.
Yeah.
There are guidance documents that say response adaptive randomization can be done in confirmatory trials.
Yes.
It's probably not very common because phase 3 a lot of times are 2-arm trials.
Yeah.
And there may not be benefit in a 2-arm trial, especially in that regulatory setting. It's unlikely that you would do RAR in a 2-arm phase 3 trial.
You have examples of seamless 2-3 trials that have done RAR in the first part and then lowered the number of doses.
Right. So Eli Lilly's treatment Trulicity, dulaglutide, it's one of the earliest GLP-1 inhibitors, was 7-arm phase 2 trial that at some point could trigger phase 3. Now, it selected 2 doses to move forward into a fixed randomization phase 3 portion. By the way, it included data from the first part in the final analysis of that, which was done through response adaptive randomization. And it honed in on the 1.5 milligram dose. Higher doses were having safety issues, high heart rate, high blood pressure. It moved away from them, and 1.5 was the dose that moved forward, eventually has done incredibly well, and billions of dollars a year treatment. So that is certainly done in seamless 2-3 trials, where the first part is to find the right dose. An interesting side effect of that is that the original design in that trial was 3 arms in the phase 2 part. And when they looked at expanding that to a range of 7 doses, that increases the sample size by that factor, 7/3, and that's too big. But by doing response adaptive randomization and honing in on a particular part of the dose response curve, the sample size isn't 7/3. And the sample size wasn't even any bigger to do 7 doses than 3 doses because of the modeling in the trial. So there's a huge benefit to that. The ICECAP is very similar to that. ICECAP was 3 durations, but they wanted a wider range of durations. They went to 10 with largely the same sample size because it hones in very quickly in the region of interest in the scenario. So the higher power isn't just higher power. It may enable more doses to be used in a better trial design.
And one thing that you talked about, well, you'd obviously talked about the Trulicity trial for a long time, but the notion that it incorporated safety, you were doing RAR not just on an efficacy measure, but a combination which allowed them to pick a dose that balanced several features. I forget what the features are. What were in that trial?
Yeah, it was a clinical utility index that was HbA1c change. Amazingly, in the world we're in, weight loss, which has become GLP-1's now approved solely for weight loss, and blood pressure and heart rate. So there are 4 endpoints that went into selecting the optimal therapeutic dose in it. Within this, by the way, we should mention, it's sort of a really interesting story in and of itself. You mentioned Don's work in this and bandit problems and its relationship to response adaptive randomization. But even going farther than this, largely, I think the first randomized clinical trial of humans was in the 1940s.
1948, streptomycin.
Streptomycin treatment of that, fixed randomization. 2-arm trial, fixed randomization. But you can go back to 1933, and there's a paper by Thompson where he introduces response adaptive randomization to that. And it was a long forgotten paper within it that has now been cited many, many times. And it's even a little bit of the search Google search things and bandit problems that has brought that paper back to life.
And not just brought it back to life. I mean, it's one of the more cited papers. I mean, talk about having to wait for fame. You publish it in 1933, you get very little attention for 70 years, and now suddenly you have 5,000 citations or whatever it's at now.
Yep. So bandit problems is largely, you've got multiple arms to pull, and you decide on which arm to allocate to a patient. Usually they're done in a deterministic way. Oh, give them arm 3, give them arm 1. And usually you've set up a goal of that particular trial, which can include a very large horizon that at some point you have to pick a treatment and go with one treatment. But you set up a goal to save as many particular patients or have a particular outcome. And this was my father's dissertation work, was bandit problems within it. So closely related to response adaptive randomization is this whole literature of bandit problems, which have also been cited many, many times. And many of this is now advertising. Online advertising, if Google has multiple ways in which it can present an ad to you, and I'm using Google just as a broad class of things, it can throw out 3 and find out how many clicks does it get. Well, maybe we'll try 2, maybe we'll try 1. Maybe we'll personalize it to individuals within that so they can be using this technology to increase the number of clicks. Okay. Now, within the properties of this, platform trials have all brought out a really interesting part to this. So platform trials are those where we have multiple agents in the trial and a relatively new thing. And we might have 4 experimental treatments and a control in the trial at one time. So this opens up the interesting question of, do we want to do response adaptive randomization platform trials, which largely are a multi-arm trial?
So I think platform trials to me are, we still haven't explored this in as much detail as we would like. We've done simulations for our own trials and so on. But RAR, it's a really different beast in platform trials, or at least it can be. If I'm doing an umbrella trial where I've got 4 arms, I've got to enroll 200 patients, when I do RAR, if I'm increasing allocation to certain arms, I'm decreasing allocation to others. In a platform trial that's perpetual, so I'm going to do 4 arms, I'm going to replace an arm when I drop it, increasing the allocation doesn't necessarily lower sample size. It slows things down. And so you could say, I'm going to speed up the arms that look the most promising, but I'm not going to abandon everything else. It's still there for me to get to it later, potentially. And I've been really interested in how all that plays out in practice. And we've done this both ways, but I think this is one of the open research areas, is how this works.
Yeah, one of the controversial things, or the things that may be problematic, is if there's 4 sponsors that own, 4 separate sponsors that own the drugs, and we accelerate sponsor A, it slows down sponsor B by definition. And that may be an undesirable thing to recruiting arms in a trial. So many of them in that scenario, phase 2 setting, don't use response adaptive randomization because
it would even be worse if we said we weren't going to explore an arm at all, much less than slow it down. That often generates more controversy with sponsors with reason. Hey, we want you to give us an answer.
Right. One trial where this was done with multi-sponsors and it was generally perceived by all to be a good thing is the ICECAP trial. So the ICECAP trial is a phase 2 trial in neoadjuvant breast cancer, and there would be 20% fixed on the control, and then the remaining 80% was set up across the experimental arms that were in the trial, and it would increase the probability. The really interesting thing there is breast cancer. We understand heterogeneity of disease in breast cancer better than most diseases. HER2 breast cancer is HER2 positive is a different breast cancer than HER2 negative. So the trial was enrolling hormone receptor by HER2 status. It also had mammal print status. So there's 8 types of cancer that women are being enrolled within that. So if you came in with HER2 negative, hormone receptor negative, triple negative disease, you have a different randomization probabilities over the 4 arms that are in the trial than somebody who's HER2 positive, hormone receptor negative within that. So we might accelerate one drug for one type of cancer, but a different drug is being accelerated in another type. And they had a fixed 120 patient sample size. Yeah. So they got more patients within the subtypes where they were doing better, and they might have gotten less in other settings. So you were treating patients better, but the sponsors perceive this to be a positive because of the multiple subgroups where RR was done different by subgroup. So it wasn't just slowing down one for the other. One slows up one place and speeds up the other place. That was thought to be a very positive thing.
Well, and it speeds up their phase 3 because if you know where your treatment has the biggest benefit, you can design a smaller trial to detect that in phase 3. You enroll the people who benefit and you don't dilute the effect through people who don't. And obviously, the people who don't benefit can go to other trials where they may achieve benefit and benefit them there.
So one of the places where I've run into the most controversy is, I've been involved in multiple dozens of trials that use RAR. Very happy with all of them. I'm really very happy with every use of RAR. We did have a couple issues in the REMAP-CAP trial. So fair to sort of point that out. These have been made public, where REMAP-CAP is multifactorial. So a patient that comes in could be randomized to drug A, yes or no, and then a different domain of treatments that you could get drug 1, 2, or 3 within that. And, I think the most a patient's been randomized is 7 different domains, simultaneously randomized that. So it uses response adaptive randomization. Some of those domains are 2 arm domains. There's a control, and then there's yes, you get therapy. Should you get high dose vitamin C, yes or no? That was one of the domains in the trial. Now, so it was deemed this is a trial that was treating a pandemic, COVID-19, and I'll be specific to that. We're also enrolling non-pandemic community-acquired pneumonia within that. But within the pandemic, it was thought that this was highly beneficial to do response adaptive randomization. It was more likely that groups that there would be a positive to entering into this trial if it was using the most up-to-date information. It's treating the pandemic while it's learning at the same time. Now, we did end up in a 2-domain case of simvastatin where it ended up about 90% probability for simvastatin versus no simvastatin. And it hovered around determining efficacy, and it probably took longer for it to declare efficacy than it would have if it stayed 1-to-1. But meanwhile, it was increasing the probability of patients getting simvastatin during the pandemic. So this was a case that was debated a lot as to whether RAR was a good thing or not in that particular scenario.
So there's another example in addition to the pandemic. We have a trial called Prospect, which there's a protocol paper out. The results paper is not out yet. We're hoping that will come relatively soon. But anyway, it is a 2x2 factorial experiment. So technically there are 4 arms, but you could think of it as doing 2 arms twice on the rows and columns of that 2x2 table. Yep. And there were similar discussions there over Prospect is on respiratory distress in children. And so the notion was, hey, we want to treat these kids well while we're trying to learn. And that was exactly part of the debate there.
Yeah. One other case, just to be upfront, is we did have a domain where initially data were reported and flipped. Yeah. And so what happens is the RAR, intending to improve the better arm, went the wrong way within that. A number of lessons learned. Now, the final data that went into it was all fixed and all of that. So, by the way, there's a strong operational burden upon, I don't want to say strong, there's an operational burden upon doing RAR, making sure the data's right that goes into the RAR, the various pieces of this. So an important part of the story is the logistical part of doing RAR. Some settings, it's reasonably straightforward. iSpy 2, it ran weekly, automated, but you got to get the data right.
I remember doing the race trial, and I would get data every 12 patients. And this was back in the old days. And so I would be sitting there. I basically put together the randomization table, and it got replaced in the randomization system. But I was more or less manually doing that, which was not what you'd want if you can avoid it. I think everything worked out fine in this, but it's been good seeing more and more groups learn how to do this and be able to do this automatically. But as you said, there are still some snafus that occur, and you want an experienced group doing this, not just on the design side, but on the implementation side.
Yep. I do want to come back to something you said that I think, in sort of looking forward to the future here, I think platform trials, multi-arm experiments are growing in popularity, even in comparative effectiveness. I do think somewhat of the future is a learning healthcare system, where if you imagine where we are today, we do these experiments to learn the right therapy, but 99.9% of people are treated outside of those, and we don't learn from them at all. As we start to merge learning about different treatments with treating patients at the same time, we do experiments where the Belmont report lays out that it's okay that these people are being experimented on, that we do 1-to-1. But in a learning healthcare system where we want to treat patients better and learn about therapies, response adaptive randomization is an incredibly powerful tool. If I owned a healthcare system and there was a particular treatment and there's 5 available drugs to treat that, IBS, psychiatry, a number of scenarios, randomizing patients to the different treatments, response adaptive randomization, learning the right therapies. Otherwise, we don't learn from these patients at all in a randomized causal way for sure, is an incredibly powerful thing. So I think response adaptive randomization, well, it's a powerful tool in the right setting. It's a bad tool in other settings. And that's the part of it, is it has a really strong future as I think healthcare becomes more integrated, learning healthcare. I think it's a powerful future.
And you're going to be exploring combinations of therapies. All of this is going to go together in ways that typically aren't done in, say, a 100-patient trial. So just because I can't.
And like the I-SPY2 trial, we're going to have a much better idea of how diseases are similar but different, heterogeneity of treatment effect. All of this comes together where I think response adaptive randomization is going to be even more valuable. Thompson is going to, post 100 years, get more and more citations as time goes on, which I think is Thompson sampling, it's even called.
Certain types. Yep.
All right. Well, it took us 60 plus episodes before we got to response adaptive randomization. I suspect it's not our last discussion of this, but one of my favorite topics and one of them that keeps people listening. So thanks for joining, Kert. Appreciate everybody out there tuning in today. Until next time, we'll be here in the interim.
Thanks, Scott.